Hello guys, today we have discussed about the Sanger model of DNA sequencing. The Sanger model of DNA sequencing is developed by the Frederick Sanger and he is a biochemist. He was developed the Sanger modeling of DNA sequencing in 1975. He was explained about the to identify DNA sequence. The Sanger model of DNA sequence is also called as chain termination reaction or terminal deoxy dideoxy sequencing method. The requirements which we have required is DNA polymerase enzyme. For example, tag DNA polymerase. Next one, primers. Next, 4 DNTPs. The 4 DNTPs are DATP, DGTP, DTTP and DCTP. Here A refers as adenine, G refers as guanine, T refers as thymine, C refers as cytosine. It is deoxyadenine triphosphate. It is deoxyguanine triphosphate, deoxythymine triphosphate, deoxycytosine triphosphate. Then next we need a template DNA sequence. Next we need the important thing in this process is dideoxy NTP. Here there are 4 dideoxy NTPs are there. They are dideoxy. N means nucleotide. What is a nucleotide? Adenine triphosphate. Next one, dideoxy thymine triphosphate, dideoxy guanine triphosphate, dideoxy cytosine triphosphate. These are the dideoxy nucleotide triphosphates which are used in the Sanger model of DNA sequencing. These are the main thing. Then can you ask that what is the difference between DNTPs and DDNTPs? We can say that if the sugar molecule as at second carbon we had no oxygen group, so it has dideoxy. It has single, so deoxy means absence of oxygen. Here at the place of second carbon there is no oxygen, so it is deoxy. In another sugar at the third carbon, here you are having which group? You are having oxygen group. But in Sanger model of DNA sequencing, absence of oxygen at third carbon also, then the sugar is said to be as di means 2, dideoxy. Deoxy means absence of oxygen. Here dideoxy, 2 oxygens are absent, so dideoxy nucleotide triphosphates. By these requirements we can explain about Sanger model of DNA sequencing. First of all, we have to take denatured DNA and it will be placed under PCR polymer chain reaction. It will be used replication and forms more DNA or larger amount of DNA. So after that, the template DNA, if we take any part, a specific part of DNA, then the DNA is called as template DNA. Template DNA, it means we have take this template DNA, it is as template DNA. It has a sequence ATGC GC. We have to take template DNA and add primers to the end of DNA. We have to add primers. We have to add primers. Then in this Sanger model of DNA sequencing, the scientist was take 4 test tubes or 4 beakers and added these primers, DNTPs, dideoxy NTPs and electrophoresis. They are added into 4 of these. After they saw that, first of all we have to take this first test tube. First we have to take template DNA. Here we have to take sequence AT. For example, I am taking it ATGC sequence, ATGC. First of all, we have to take here we have dideoxy ATP. Here we have dideoxy TTP, dideoxy GTP and dideoxy CTP. We have to take first tube 1. What is dideoxy ATP? If we take, what is the opposite of adenine? It is thymine. What is the opposite of thymine? It is adenine. If we take dideoxy ATP as opposite to it, complementary to it, then dideoxy ATP, then it will be stopped. Here firstly we have to say that it is a chain terminal dideoxy sequencing method. Here we have to say that if the dideoxynucleotide triphosphates were added to the DNA template strand, then the termination will be occurs and the nucleotide primers will be stopped. There is no elongation will be occurs. So when the dideoxy complementary adenine will be added to it, then the termination will be stopped. Then what is the nucleotides are there? There is 1 and 2 on it. Then if we take second time, what is the complementary to adenine? It is thymine. Then what is the complementary to thymine? It is adenine. If we here we have DNTPs also there. DNTPs, DN ATP, DN TTP, DN GTP, DN CTP. If we take DNTP as here, then the thymine will be occurs. Then next guanine opposite is cytosine. Here I will say that A is complementary to T, T is complementary to A. Then guanine is complementary to cytosine. Here triple bond will be occurs to guanine to cytosine. Here we have to take cytosine means guanine, adenine means thymine. Thymine means we have to take if the dideoxynucleotide will be occurs, so dideoxy ATP will be occurs and it will be stops. So what is the number? 1 2 3 4 5 6. The 6 will be occurs. So if we take second tube, then take one strand. What is the complements? AT GC AT GC. Then what is the complementary strand to it? Here is dideoxythymine to it. What is the adenine complementary? Dideoxythymine. So dideoxythymine triphosphate. So there is only one is there. So next adenine complementary. Here if we take thymine to it, so adenine complementary thymine, thymine complementary adenine, guanine cytosine guanine. What is the opposite to it? Adenine thymine. If we take these dideoxythymine, so it will be stops, terminates. So what is the number? 1 2 3 4 5. It is 5. Then if we take third tube, it is guanine, dideoxyguanine. So we have to take template strand ATGC ATGC. Then what is the complementary of adenine? It is thymine. What is the complementary of thymine? It is adenine. What is the complementary of guanine? Cytosine. What is the complementary of cytosine? Guanine. Here is dideoxyguanine triphosphate is there. So dideoxy GTP. So it will be stops, terminates. So 1 2 3 4 are there, 4 nucleotides. So if we take another time, so what is the adenine? Thymine, adenine, guanine, cytosine. Here we take cytosine means guanine. We take D and GTP. So here G will be occurs. So A means D, D means A, G means C, C means DD, dideoxy GTP. So here we have to see 1 2 3 4 5 6 7 8. 8 will be occurs. If we take fourth tube, ATGC ATGC. What is the complementary of A? T. We have to see that dideoxy CTP. What is the complementary of T? A. What is the complementary of G? It is dideoxy CTP. So dideoxy CTP will be occurs. It will be terminates and stops. Here we, what is the number? 1 2 3. Here will be 3. Then if we take another step, A means thymine, thymine means adenine, guanine means, here we take D and TP. So D and CTP. So here we take cytosine. Then cytosine means guanine, adenine means thymine, thymine means adenine, guanine means cytosine, guanine means cytosine. Here we have to take dideoxy CTP. So dideoxy CTP. So it will be terminates 1 2 3 4 5 6 7. 7 will be occurs. So these strands will be go under electrophoresis. These tubes will be, 4 tubes will be placed under electrophoresis. Here we have to numbering from bottom to top: 3 4 5 6 7 8 9 10. Here 4 tubes are there. Tube 1, tube 2, tube 3, tube 4. Here what we had taken in tube 1 dideoxy ATP. In tube 2 dideoxy TTP. In tube 3 dideoxy GTP. Tube 4 dideoxy CTP. We have to place these numbers here. So what is the number occurred in tube 1? 2. Here we have to place it 2 and 6. In tube 2 in dideoxy TTP it will be occurs 1 and 5. It is a 5, it is a 6, it is 2, it is 1. Then tube 3 will be occurs 4 number. Here will be 4 and 8. If we take tube 4, then it will be 3 number and 7. By these we can be give a complementary sequence of that. Then what is the one number? T, thymine. Then second number is adenine. Third number is cytosine. Fourth number is guanine. Fifth number is thymine. Sixth number is adenine. Seventh number is cytosine. Eighth number is guanine. It is the complementary sequence to the template strand of DNA. What if for example we have taken ATGC. For this DNA template strand, this was the sequence, complementary sequence for sequence: adenine, thymine, adenine, guanine, cytosine, guanine, thymine, adenine, cytosine, guanine. By doing this dideoxynucleotide triphosphate process, we have to give a complementary sequence to it by doing Sanger's method. By termination of third carbon of dideoxy, we can give a complementary sequence to it. This was the Sanger's model of DNA sequencing. If you understand this process or this method, please subscribe to it and share to your friends for examination. Thank you.